in this audience, everyone, of course, is familiar with Star Trek, the original series. And when the crew wanted to determine what kind of life forms were available on a planet, all they had to do was whip out the tricorder, and they instantly knew everything they had to know, and the plot could progress quickly. Here on Earth, things have not progressed as quickly. 2,000 years ago, we had Plato talking about his featherless bipeds, and 300 years ago, Linnaeus moved taxonomy on a little bit, and today we've got about 1.5 million out of the 10 to maybe 100 million species classified. But it's taken us a long time to get there. And now, thanks to these gentlemen, we finally have our tricorder. We have the ability to take this little machine, put some sample tissue in, and get a unique species ID out. They've done 30,000 species so far, and have a proposal to do 500,000 species over the next 5 years. And note that that's a third of what we've accomplished in the previous 2,000 years. And so we have our tricorder. Let me introduce you to our Spock and Kirk. I'm not quite sure which one's which. But Dan Herbert, who proposed barcoding in a 2003 paper, is chair of zoology at the zoology department at the University of Guelph in Canada, a fellow of the Royal Society of Canada, and many other honors. Dan Janzen is a Demory professor of biology at UPenn, National Academy member, winner of the Einstein and Kyoto Prize and MacArthur Fellow, among other honors. And they'll be here to tell you today about all about barcoding of life. But before that, in addition to Spock and Kirk and the tricorder, there's one more thing you need to explore a planet, and that's the away team. And Google has proposed, with the help of these gentlemen, to do our own away trip to the jungle in Costa Rica. And that'll be on the weekend of March 28th, and we'll be able to take about a dozen Googlers. So you'll be the red shirts. And don't worry, historically, at least half of them come back alive. And if you're interested in that, in going into the jungle and learning about the biodiversity there, you can sign up in Jonathan Rosenberg's office from 2 to 3:30 today. So stop by there, make your case for why you should get to be the red shirt. And now we bring you Dan Herbert, and Paul Herbert and Dan Janzen. Thank you.
Thanks very much, Peter. Well, it's a real pleasure to be here, and I want to thank you for the introduction, and Jonathan for the invitation, and our emissary to Google, Brad Slotnick, who put a lot of time into helping us have a chance to speak with you. For the last 3 billion years or so, organisms have relied upon analog gradients to recognize the life forms around them, to decide who's friends, who's foe, who's family, who's enemy. And so if we look at this kaleidoscope of life here, you're recognizing the differences by looking at an analog gradient using sight. We're proposing that that system works rather well with small assemblages of species, but as the number of species that one is dealing with rises, it runs into problems, and that we have to look at creating an identification system for life that's based on a digital character set. And so here you see a DNA barcode, a 650 character line of DNA sequences. And for the balance of my talk, I'm going to be focusing on the evidence that this approach works, and just talking to you a little bit more about the problems and the opportunities that come with creating a DNA-based identification system for life. So DNA barcode, what is it? It's a short system. It's based, we're trying to develop the most parsimonious system possible to tell organisms apart. A single DNA read is the cheapest currency in our business, and 650 base pairs is what current technology will deliver. Standardized. It's critically important that we focus on a single piece of DNA across the spectrum of life. If not, you've got to know if you've got a beetle in your hand or a fish in your hand before you could select the gene region. So the idea is to standardize on drive a single genic railway gauge through life. And of course, we'd like that standardized sequence to enable species discrimination in a large block of life. Wouldn't it be magnificent if you could walk to the seashore and that mollusk, that cnidarian, that algae, every one of those organisms, you could gain a species identification simply by reading a small slice of the DNA in the organism's genome. So here's an example. A wood duck, a honeybee. Yes, you can tell them apart. Those are their barcodes. You might argue that, well, can I tell them apart? Just look at the color spectrum along there. You can see there is a shading difference, right? The wonderful thing about these 650 base pair characters, of course, is they can be automated. They can be read with automation. The basis for the idea behind barcoding lies in the simple power of combinatorials and the fact that DNA is basically a digital DNA string. We're all familiar with the basic barcodes on store shelves. 11 long, 10 options at each, a huge number of combinations. But DNA is a combinatorial string as well. There's only 4 options at each position. Those are the 4 nucleotides that go to make up DNA, the 4 different nucleotides. So you can read along this string, and of course, 4 characters at each. If you even read just 15 different nucleotide positions, you get 1 billion different combinations. So it seems like we might not need to look at much DNA to recognize the different life forms on the planet. So the power of combinatorials, DNA is immense long string in your genome. If we were to count the nucleotides, there are 7 billion of them. We could actually take this to a very high power if we had it to. But of course, taking it to a high power would mean lots of DNA sequencing and great expense. So this is a barcode for this little fly. Legs away for DNA. 1 leg came off this fly. The fly is still intact. You don't need much of the organism in order to gather a barcode. In fact, you need just touch it. And these are difficult words to pronounce, I will have to say. They're even worse than pronouncing some of the names of Welsh towns, I would argue. 650 letters long, but they're simply codified. An N is where one didn't get a character read. There was a sequencing problem. We shouldn't show sequences like that, should we? That's the harsh reality. You always can't read the sequence perfectly. Why should we really care about the ability to inventory life on our planet? There are a lot of social and economic reasons why we might want to do this. Invasive pests. California had an invasion of the Mediterranean fruit fly a few years ago and spent $150 million eradicating it because that pest had got a fairly strong foothold. Zebra mussels got into Europe from Europe to the Great Lakes, $500 million damage in the 20 years they've been there in our country, and it's spread down through the States. And large, billions of dollars can be, a single invasive species can cost that. So it's an invisible tax on society. And in a world where we're globalizing trade, where climates are changing, we're going to see a lot more species invasions. A good biosurveillance system will allow us to stop these organisms at the beachhead and to save vast amounts of money. If you go to a restaurant and have a fish meal, at least in New York City, you've got about a 20% chance of not getting the fish that you paid for. It's always an upgrading. A low-value fish will be substituted for a high-value fish, and not that many people can tell a fillet apart on a plate. The FDA is looking at using barcoding as the method to surveil foodstuffs in the marketplace in the United States. 62% of the emerging diseases that are affecting our species use another organism for transmission. They're zoonotically transmitted. A number of organisms that attack humans, including the malaria that mosquito might be injecting, are eukaryotes as well, and would be DNA barcoding could give you a fast identification of them. Fungal diseases, parasitic worms that are not trivial in much of the developing world. And then here's a specialized application. If you're into aircraft design, it's often useful to know what size bird ricocheted off your aircraft and caused a dent in the aircraft. It might cause you to shift the flight pattern, or it might cause you to change the design of the aircraft so that the bird didn't come through the aircraft fuselage. And the FAA is supporting barcoding because they got tired of looking at smears of red on an aircraft and trying to reconstruct what the bird was from the blood. But of course, with barcoding, that's straightforward. And we live in a planet where humans don't always treat the environment kindly, and it's important to be able to monitor the environment. It's often very difficult to identify the organisms that are used in biosurveillance programs. And so both in Canada and in the United States, the EPA are investing in barcoding as a new generation tool to surveil environments. And of course, this is just the front end of this approach. It can get, we can begin to use highly sophisticated biomonitoring to look at the planet. Just as canaries were dumped down mines in the 19th century, we can surveil the planet. Not just measure wind speeds and temperatures, but the life forms at each spot on the planet. The other thing that is really, I think, a moral responsibility. 65 million years ago, a 100-meter diameter meteorite impacted near the Yucatan Peninsula and took out 75% of the species that were on the planet at that point in time. And of course, the dinosaurs hadn't planned on it, didn't recognize what was happening. Today, the situation is rather different. We as a species are preempting 25% of the primary productivity on land today. And most of the people in my business believe that we could be responsible for the loss of 50 to 75% of the species on our planet's surface within this century unless we change our behavior patterns. These are fellow travelers for millions of years. And to think that we would be responsible for eradicating them with no record is, I think, something that's not a trivial issue. These are 36 of the 37 members of the cat family on our planet. There's only one of them that's not threatened with extinction, the domestic cat. It could be the only cat species in the wild by the end of this century. That's just one biological microcosm. Every group of life on land is highly threatened. So let's jump back in time. Let's jump back 250 years because this is the quarter millennial celebration of the scientific registration of life on our planet. Linnaeus began this task in Sweden in, well before 1758, but the 1758 edition, the 10th edition of Systema Naturae, is when biologists say we began to register species in an official fashion. And Linnaeus, in that document, registered 5,000 species. And in the case of butterflies and moths, he had about 500 species of butterflies and moths registered. He thought he had half the job done. He felt there might be 10,000 species on the planet. Well, Peter mentioned it's not 10,000 species. Today, we're at a count of 1.6 million species registered and many specimens residing in museums. Collections like this, magnificent repositories of life. But we know the job isn't done. It's very likely that they're in the tens, if not hundreds of millions. I can't give you an order of magnitude closest estimate after 250 years of work. A few years ago, I joined in a Bio-Blitz in the Great Smoky Mountains National Park. That's one of the best studied and most visited parks in the eastern United States. And these are the sites that people assembled to collect. And this particular mission was focused on learning something about the moths and butterflies of the Great Smoky Mountains National Park. Whoops. These are the people that came together to do it. 30 individuals assembled with special understanding of different groups of moths and butterflies. For 3 days, there was a powerful bioidentification force on the Smoky Mountains, and then they all dispersed, and the capacity went back to zero. And of course, that team could not deal with the multitude of other groups of life. It's extremely difficult to deal with the diversity of organisms with the traditional approach of morphology. In fact, it's this disconnect that's made rather graphic here. Our planet home to somewhere between 10 and 100 million species, and the human CPU able to deal with perhaps 1,000 of those species. So if you decide to devote your life to something important like taxonomy as opposed to Googling, well, of course, we'd like to see a confluence of taxonomy and Googling. You might be able to get to the point where you could discriminate 1,000 species, but it probably will be at only one life stage, just the adults, not all the larval forms. So just a segment, your life, 1,000 species. This vast disconnect is why we need to look for a new approach. We need to do what the other sciences have done, which is bring technology to bear on the fundamental particles of their discipline. And that's what barcoding is all about. So we're engaged in the early phase of a mission that seeks to carry out a horizontal genomic survey. The big genomics projects you've heard of in the past focus on assembling information on all of the genes in specific species, our own species. 2001 was the early closure of the human genome. This is a very different enterprise. This is orthogonal to traditional big genomics projects. We want one gene. In fact, we want one gene region from every species on the planet. We want to build this reference library connecting a sequence to every species on the planet. This looks a bit like a stellar image. Well, it is a stellar image, but it's not a space image. It's an image of an animal cell. This is the cytoskeleton is in red. The nucleus, the huge block of DNA, is sitting there in blue. 7 billion base pairs in the case of our own species sitting in that big circle. And these little green dots These are mitochondria. These are the powerhouses of the cell. And the interesting property about mitochondria, not only is this high copy number, but the fact that they've got a little loop of DNA as a relic of their bacterial origins. And that little loop of DNA, because there aren't sophisticated repair systems in there, tends to evolve very quickly. So if you're looking for a piece of DNA with a lot of signal, mitochondria are a very good place to go. Not only a lot of signal, but lots of copies, so it's relatively easy to recover even if the fish has been cooked or the DNA has been degraded in some fashion. So we have targeted the mitochondrial genome as a point of first entry into barcoding. And this is the mitochondrial genome. It's a little loop in all animals, about 16,500 base pairs long, so it's tiny in comparison with the nuclear genome. And it turns out that, well, as I mentioned, we're ambitious. We would like to create an identification system for all nucleated life. It turns out there are 13 protein coding genes in that loop, but only 2 of them, cytochrome oxidase 1 and another gene over here, are present in all eukaryotes, in fungi, in protists, in plants, just 2 genes. We focused on one of those genes, cytochrome oxidase 1, and we focused on a 650 base pair bit near the front end, because humans like to begin at the beginning rather at the middle of something. And so that's the region that we have designated as the barcode system for eukaryotic life.
So where's the middle of the circle?
The middle of the circle? There's no such thing as a middle of a circle. There's a start point. Okay, what do we do? There's a start to a gene. There's a start to a gene. When it gets transcribed, it rips off in a particular direction. So what do we do? It's a pretty simple analysis. A human genome project, Craig Venter just got finished, and that was a $70 million project. We can give you a barcode for $2 or $3 today. So this is a lot cheaper. This is why it can be done in scale. So take a specimen, take a fish, take a tiny piece of tissue, so tiny you can't see it. Do this from 96 fish, put the little bits of tissue into a block like this, extract the DNA, all of the DNA in that cell, and then amplify this up. PCR, polymerase chain reaction amplification, basically allows you to Xerox copy. You've got these 7 billion base pairs, and you can jump in and Xerox copy one particular piece of it. Got to do that to get the barcode. So we go in, amplify up that piece of DNA, and then it just jumps into a sequencer, a capillary sequencer, and gets read the same way genome sequencing has been done in the past. Sequence record, trace files, and sequence record. The other thing we do is gather the collection data. Where was that organism collected, when, photograph it, create a specimen page. Today, barcode analysis, you're interested in doing it. We built the world's first barcode factory in Canada, in Guelph, where I'm based. We have a team of about 35 people there that devote their life to trying to make this easier and faster, less expensive. Use robotic intervention, microfluidics, capillary sequencers. There's an informatics team, and this little building can process 500,000 specimens in a year, 1 million sequences. And if we had 5 or 10 of these scattered around the world, if we franchised this little facility, we would be in a powerful position to register life on this planet in short order. One of our challenges is informatics. We're generating quite a lot of data. We need to gather those sequence files together with the specimen information, and that's what this database is doing. It currently contains information for about 35,000 species. That's a tiny fraction of the 10 million, but it's the start. This is what one of those files looks like. The taxonomy, the location data, where you can find that specimen if you're interested in looking at it again, images of the specimen, an onboard GIS system that shows you where that specimen was collected. And here's the other part of each record, the sequence information, the trace file, and the sequence record itself. That's one barcode record. This is something that you're familiar with. Google Earth's a magnificent thing to link our data into, and here we're zooming into Costa Rica to ACG, where Dan Janzen works, and Winnie Howlocks, and here you can see zooming in then to this one locality and seeing 2 specimens that were collected just off map here. 60,000 records available for the ECG today. Imagine when the whole planet is like this and there are millions or billions of records, and you can dive into anywhere on the planet and see the life that resides there. This really becomes a digital bioscope, if you like. Does barcoding work? We've tackled 37,000 species so far, and it works with startling clarity, I would say. I don't have time to show you lots of examples, but our planet is 75% marine, so marine seems a good place to begin, and Great Barrier Reef is not one of the less pleasant places on the planet. So some of our earlier barcode work was done with CSIRO in Australia, and here you can see the sort of, this is called a taxon ID tree, and I don't want to spend too much time looking at this, but 2% divergence, add that, and perhaps you get about 10% divergence. That is the sequence difference between the two most closely allied species in that collection. These lines that are straight, if you see a straight line, no variation among members of a species. Little bumps, tiny amounts of variation within a species. Our own species, every person in this room, we sequence you all as you walk out, there won't be more than one base pair difference between any person in this room. It was thought that humans were a bit of a funny species, the so-called mitochondrial Eve hypothesis. It turns out every species we've looked at shows this same sort of pattern of lack of variation within it, deep divergences between it. Now, it turns out these fish that you're looking at here, there are 2 people in the world that could tell those fish species apart morphologically. With barcodes, everyone in this room, everyone on this planet could tell them apart. I don't want to show you more examples, but I'll just say that the startling thing is, whether you're looking at a fish in the ocean or a crustacean or a jellyfish floating through the water, you will see the same sort of pattern. If you jump onto land and you look at birds, if you look at bats, bees, frogs, odonates, ants, you will see the same sort of pattern of sequence variation. The resolution is extremely high. So we're now engaged then in building this reference library that connects sequences to all of the known species on the planet. And what we're going to get from that is a master key to life that anyone will be able to open and look at the diversity of life on our planet. The other nice thing about DNA is genders are the same when it comes time to barcode region. If you're looking morphologically, genders often look different. The identifications are cheap. $3 today, it will be a few pennies before long. 2 hours today, it will be a few minutes before long. And the other nice thing is that DNA barcodes are constant throughout life. So in the case of this little marine crustacean, the adult, which taxonomists can tell apart, connects to a whole kaleidoscope of larval stages that look hugely different from it. Think about cat egg, caterpillar, pupae, and adult butterfly. This transformations through life complicate morphological taxonomy, but not a DNA-based approach. So, based on these early results, what we're at the stages of now activating is the International Barcode of Life project, and it seeks to gather information on 5 million specimens, 500,000 species in 5 years for $150 million. That's a tiny price in terms of big science projects. Fundraising, 3 models, central nodes, Canada, United States, and Europe playing a heavy lifting role, and I'm happy to say that Canada's indicated that it will commit $50 million towards this project, but we need international partners. We'll gather the species from charismatic sites, Great Smokies Mountains, Banff, Iguazu Falls in Argentina, magic places to capture the attention of the public, the ACG in Costa Rica. We need help on informatics and data display. That's a big challenge for us. Here is the future in terms of technology. Go to a tree, fog the tree, put it in a blender, create a slurry, amplify the DNA in that slurry, put it on one of the new generation sequencing machines that sequence 400,000 sequences a day, and read the life. Go to Amazonia where the forests are being slashed, fog trees in each of these areas, and read the life. Make decisions that are scientifically based on which areas to protect. We can't do it today. I wish we had the tricorder. We don't. That's what you guys are going to create for us. No. This little device is what our holy grail. We really want a handheld device. It's not going to be just point. Going to have to touch it. Going to have to wait a minute. But that little slot there, touch the frog. Put the piece of paper in that slot. PCR amplify, sequence, match against the reference library, identification, Linkia lavigata. Are you holding a purple starfish in your hand? Are you on the Great Barrier Reef? No issue. If you're in the Caribbean and you encounter this thing, it's invasive. Reality check. We had a meeting in June, Canada. 75 people came together from 21 nations. We can do it. So, this is what the International Barcode of Life project is about. Imagine a world in which you can know the name of any animal, any plant, any fungus, any organism, on the spot, in an instant, anywhere on our planet. We can, as a society, do this in 5 years with a very modest investment. The end of this, we've created a global bioidentification system. This is biology's answer to global positioning systems. Global positioning systems revolutionized our ability to put ourselves in a point on our planet. This approach will allow us to position ourselves in taxonomic space in the organisms around us. It's going to be critically important for life on our planet. We would develop this approach so that we can do our best to protect organisms. And then finally, society should be very engaged in helping to pay for this, but because we're going to save society through the interception of a single pest species, more than the cost of this whole project. So that's my entree into the project. Dan's going to jump up now and I'm sure give us a very interesting, different perspective. We're allies in this.
We have about 100,000 years of knowing about biodiversity and forgetting about biodiversity as a species. It goes back further, of course, but as conscious human beings, let's call it, we've been thinking about it for a long time. There's you as an individual right now, and of course there's about 10 million species. Whoops, where's the pointer on this guy? It's in the middle, isn't it? There we go. There's about 10 million species out there. And your problem right now is you want to know the name of that frog because he just stuck his finger in your eye. I don't jest, actually. That frog stuck its finger in my eye when in 1961 in southern Mexico when I wrote my first scientific paper about what happened. Any 8-year-old today will tell you how you get the name and understanding of that frog is just to go to Google and type it in. No problem. You're all done. Well, the real problem, of course, is how do you know which of those 10 million species goes with that frog? And you just heard Paul talk about it. Okay? That's where we're headed. That's how I see it as a user at the bottom end of this feeding chain with Paul sitting up there in his barcode factory in Guelph at the other end. So this is the sort of dream slide which says in one slide the same thing you've just heard from Paul. And that has its origin in thinking this way. The idea, of course, is that there are 7 billion people in the world who can pull something out of their back pocket that costs what this costs and is as available as this is to do this job that we're talking about. That is really where we're trying to head. Now, you say, well, yeah, you know, I can get some taxonomists to tell me the name of this frog and so on. I am standing in this photograph in the middle of a piece of Costa Rican rainforest, and those of you who go with us at the end of this month will be put in that forest. There are 400 species of plants in that photograph. Every one has had a name for 50 to 100 to 150 years, and there is not one human being on the earth who can stand there and identify more than one or two of them. You can collect it, send it off, 6 months later, get a name. But if you have any reason whatsoever to want to know what the name of that plant is, nobody else can do it unless you pay the salary of somebody to come and stand on your left hand, and he will only know about 2 or 3 or 4 of those. This is the level of our understanding of wild biodiversity. This is your visual. This is what you see when you look at wild biodiversity. And we're trying to do something about that. Now, let's take it right down home pragmatics. This is your arm somewhere. We all know what's going on there. And what is your reaction to this? Well, that's your reaction to it. Okay, now you've done that. And you wonder, could it have been carrying malaria? Could it have been carrying yellow fever? Could it have been carrying sleeping sickness or some trypanosome or some other horrible thing? Or was it just an inconvenient itch? The only way you can know is have a barcoder, take a leg off that, and go through the same process that Paul was just talking about. This is not a setup photograph. It's not an exaggeration. This is the world in which we are now developing. We're joining the most primitive, down to nature, with the most advanced technology that humanity can put on the table. But it's got this problem. You see, there's no little hole in that gadget right now to put a piece of the animal that he's got and standing on after he just shot it. We all intersect with wild biodiversity at some time in our lives for some reason. Not all the time, that's the professionals who do that, but the 7 billion people just now and then need to know who the species are. You thought you ate lobster for dinner last night. How do you know, as Paul just indicated, it wasn't Nemo that you ate for dinner last night? Well,
with the iPhone's pinch feature, you can zoom in on pictures.
there it is. There's the barcode right there.
Whoa.
Now, if we drop down into the real world, let's take a look at real complexity that you get confronted with when you're not out here in a city park in Palo Alto. That little yellow lump right down there is Costa Rica, and you can see its relative size up against the map up there. The same number of species are in this little lump down here as there is in all of North America. So what I'm saying to you is you're really packing species into a little tiny place. And so there was a whole lot of them every time you turn around. That's where that piece of forest was we were looking at earlier. Now, if you apply a barcoder to this kind of complexity, how really accurate is it? How well does sequencing work for this kind of identification? Well, right now in this place, we have done 3,000 species of moths and butterflies, 700 species of flies, and 600 species of parasitic wasps. And I have to add, these are all things we have reared, so we know their whole natural histories. We know the animals very well. And out of these numbers of species, 7 didn't work, 2 didn't work, and 6 didn't work. That's the error term in this gadget. Now, furthermore, they didn't work in a peculiar way. Those 2 right there, it isn't that they're confused with anybody. It's that there's 2 species you can't tell apart this way, but they don't confuse with any of the other 700 species of flies. Okay? That's what I mean by it didn't work. The point is, it's extremely accurate. Now, let's look at the world you know in relation to this kind of thing. This is Flickr, which you guys all know. And I put a name of a butterfly that I happen to know into Flickr right there, and I immediately get these photographs. Well, let's look a little closer. There's the picture of the butterfly. He's feeding on a rotten banana in British Honduras. Some tourist took this picture and put it up on, actually he's a professional photographer tourist, and put this up on Flickr. Hey, proud, you know, see my butterfly. And some taxonomist said to him, yeah, that's Laertes propona. He actually wrote it on wrong. It's actually propona laertes, but that doesn't matter. So I put that butterfly into your search engine, and there it is, and you get 638 hits. So here's a block of information about this butterfly. Notice no barcoder involved here. A block of information about this butterfly straight out of your search system. You think, wow, we have a fire hose worth of information about this butterfly. Well, I've reared these things, so I barcode them. When you barcode them, that's the butterfly right there with his wings open. Here's the caterpillar that's reared from. And you barcode them, you discover there's one group here. These are all individual butterflies. And there's another group right there. And they don't have the same barcode. And then when you look at this group, you discover they're all eating one family of plants, and these are all eating another family of plants. That's 2 species. And there is not a taxonomist in the world who can tell them apart. And they've had that name for almost 200 years. Every single one of those 683 pages in Google is wrong, because you have no idea which one of these two any given set of information goes with, because they completely co-occur. These 2 butterflies live side by side, same 1 meter apart. They can tell each other apart, but nobody else can, except somebody with a barcoder. Now, I use Guelph as though I'm carrying it around in my back pocket. Okay? This is 9 species of flies who all have the same name. That name's 150 years old. Everybody thinks that's all one fly. It's in fact 9. We know that because we rear them from different, they're all parasites of caterpillars, and we've reared them from different, when you barcode them, they all have different barcodes. The only way you can tell them apart is with a barcoder. There are 10 species of butterflies in this box, all from the same place, all reared. We know all of them. 6 of them have had names for more than 100 years. 4 of them are unknown to science, and there's not one living human being on the planet who can look at that photograph and identify them. And there never has been. Yet, with a barcoder, $2.50 a piece, you can unerringly identify every single one of them. The point is, we need a barcoder to find out what we already know, and then when we barcode what we already know, we discover that we didn't actually know what we thought we knew. So the world is more complicated, excuse me, than we thought. So where do you get this information? I'll take you right down to the ground level. Where does the sequence come from? These are what are called parataxonomists. The word is borrowed from the word paramedic, who drives the ambulance. She is Hazel is the wife of a school bus driver in northwestern Costa Rica. Sergio was a taxi driver up on the Nicaraguan border. We plucked them out of these very working class jobs, taught them the things you teach a good graduate student, and they're out there being parataxonomists, gathering specimens explicitly to create barcode library. What Sergio's doing is blowing up a plastic bag to put a moth in it. It gets a clothes fit on it, goes in the freezer, comes out of the freezer, gets put on a spreading board, goes into an oven, is dried, comes out looking like this, butterflies and moths on pins, excuse me. Each one of them gets its own individual license plate, a voucher code that goes with it. They photograph it, they build a record and a database for it. That all goes on the web, of course. One leg comes off, goes in a little tube right there. The boxes could be FedExes to Canada, or in this case, a mule takes them to Canada. They get to Guelph, the leg comes out of the tube, goes through the fancy robots, comes out as sequences. Now, we have a barcode library building. Who's going to do that? We have 20 people doing it in northwestern Costa Rica, and there's people in the Great Smokies doing it a little bit, and so on and so forth. But what's the army of people who can do it if you suddenly put gasoline into the motor to get 10 million sequences for the world? Excuse me, the sequences from 10 million species for the world. Well, there are a very large number of people who, for one reason or another, deal directly with biodiversity. That is the only human being in the world who can identify one big set of flies, for example. Well, they occur in very large numbers. In fact, by my off-the-back-of-the-napkin calculation, there are about 100,000 people out there who deal on a daily basis with species for one reason or another, who can be enlisted to be part of the army who gathers this whole barcode set. It isn't that I or Paul or one graduate student have to go out and then spend the next 2,000 years doing it. The army is out there. These are all experienced biologists or biodiversity managers or people using biodiversity in one way or another, and they will in fact do it if they have the right administrative structure. Now, what does it look like when you actually get these results back from Guelph? You know, they or us, we send this leg off and we get this tree back with information on it, and right there is one that has no name at all. I don't have any idea what it is by looking at it, and it's sitting way out on its own little branch. How do I get this linked back to whatever it is we know about biodiversity? Well, I'll show you actually how we do it. The first thing you do in the old days was to take that specimen and airmail it or hand carry it to the British Museum, and it sits there for 6 months, and people look at it, and maybe out comes a name. Today, you send it to the equivalent of that in Costa Rica by taking its photograph, attaching its photograph as an attachment to an email message. It goes off at 7:58 in the morning. At 8:08 in the morning, you get a reply back. This is the genus, this is the family. Maybe somebody else there knows the name of the genus or species. And then 2 hours later, you get back the species name, where it lives, how many specimens there are, and so on. The point is, that's the old-fashioned way, but speeded up now by all the internet and other kinds of things that we have. Notice there was a barcoder only put its finger on it and said, this is a problem, it exists, and we use the old-fashioned way of getting to it. And finally, they send us the photographs of the Thai specimen and a Google map that shows us the places where the thing has been found before. Now multiply that by 10 million, okay? Now, here's a very practical example of this. This is the watershed of New York City. The black that you see right there, there's Long Island there. And New York City is very worried about the quality of the water that flows in that watershed. These are all the little streams in that watershed. So they pay a lot of money to monitor the biodiversity of those streams. Biodiversity means all these little things that live under rocks. And these are the streams themselves. Red means that they're in really bad shape. Greens means they're okay. And how do you classify a stream as good or bad? Well, you make a list of all the species that are in it, and one set of species represents somebody's been dumping their laundry water into it, and another one means it's a pristine stream. Well, how do you actually get that information today, in today's world? This is the way you get it. You go out and you sample, and you get liters of insects out of the stream, and you sit down, a whole lot of undergraduates and volunteers and some paid labor, and they sit there for hours and hours and hours and hours and hours, morphologically identifying the bug one by one by one by, and they make a long list. And literally you can spend that team like that can spend a whole month doing one stream. That whole thing is replaced with one barcoder. These are 372 legs off of known barcoded samples, so you know what the barcodes are. These, dump them in a sequencer, and you're done. It changes the whole way of going at this. So where are we headed now? The year 2020. We know about this world. It's got Google up in this corner. Put a barcoder in that corner. You've got a laptop in everybody's hand down here. And you've got the human genome, which loves to teach itself things. We all know that. When you were 2 years old, you learned, when you're in second grade, you learned to teach yourself a lot of things. Well, what happens as soon as you have that, you get all these other things begin to add on to it. EOL, which has been talked about, came out yesterday or today in the New York Times. You get instant messaging. You get Facebook pages. You get all the written documents that we own out there in the public domain, which you guys are busy doing. All that stuff works together, and it isn't going to be long. Paul skipped over this, but I will say it won't be very long before the organisms themselves are going to start to transmit their own barcoders, or their own sequences, their own barcode, in the sense you'll be able to pick it up. So what's the hurry? Why not just let the process go, and in 100 years we have a barcoder and we have a sequence library growing? That's the way we've been doing taxonomy, the way we've been doing biodiversity. Well, it goes like this. If you can't read what's out there in the forest, in the old days it was the enemy. So we push it aside, grow our crops. All right? Then more recently we think of it as, hey, if you can't read the forest, it's something you eat up and turn into firewood. And today where it is right now, it's biofuel. Ethanol, biofuel, I'm not worried about corn at all, is the worst enemy we have of the remaining living biodiversity on the planet at the present time. We don't have time anymore. Now, Who are all these people that are being impacted? You think, is it us here in Palo Alto? There are 6 billion people out there that live day to day, face to face with wild biodiversity. It matters to them now. They need this thing in their hands right now. And imagine how it would change politics, education, medicine, the whole pack, agriculture, the marketplace, everything. If these people could know what they actually have in that wagon behind those oxen, what that mosquito was who was nibbling on your arm, what the beetle is that's eating your bean plant in Nigeria. Barcoder, it's the first time in human history we can put all 7 billion people on the planet and all the 7 billion wild things on the same team, working together. That, to me, is true democratization, or bio-illiteration, as I'm inclined to say. Now, what do these things have in common? You all know what these are. And we hit right down there, we hit Google down here at the bottom. What do all those things have? What's one thing that they all have in common? There's something in it for everybody. We are not talking about something for the academics, for the graduate students, for the wealthy people, for any particular poor person. We're talking about something for everybody. Now, this slide, Van Ton loaned me as a slide that was put together with respect to the E.O. Wilson Foundation, which I'm very familiar with. And he pointed out that this is Google. This dark green stripe right across the middle here is Google. And indexing right down here, of course, that word is exactly what you guys are busy doing to all the human-created things, what the barcoder is talking about doing it to all the wild things. So I'm going to close down here by pointing out that the database for this entire information ecosystem, from teachers and students and up to public health and conservation, all that sort of thing, is 7 billion people barcoding all the species on it. That's the real bottom, the real platform of this thing. So in the end, this is what we end up with. And that's really the information ecosystem that Google is right a stripe right across the middle of, and the barcoder is the thing which is actually producing the raw information down there and available to everybody in this whole system. Thank you.
Any questions?
You mentioned that you're using mitochondrial DNA to do the barcodes for eukaryotes. What about prokaryotes?
We decided eukaryotes would keep us busy for a little while. So the question was, what about prokaryotes, which lack mitochondria? We've decided to just stand aside and focus on the 10 million or 100 million eukaryotes as the first line of business. But the idea of standardization of genomic targets and focusing on as little information as possible are principles that we could apply to prokaryotes. And interestingly, the same gene that one finds in the mitochondria of eukaryotes, of course, is present in many prokaryotes. So that would be a friendly place to begin. And occasionally, bacterial or archaeal CO1 jumps up when we're doing an analysis on eukaryotes. That's a good question.
Let me add something to that, though. The concept of barcoding, we're focused technically on the CO1 gene because that's a technical target. But the concept of having a signature sequence applies to anybody. And the medical world is already there. I can say freely, this is public information, that Canon is developing a gadget that you put on your desk. It'll cost you $5,000. Your daughter comes home from school, she's got a fever, she reaches out and touches her tongue, you have mumps. That's where it's going. And the minute every doctor has one on every desk, of course, you're going to want to have one in your own kitchen. And so that they're focused on all the human diseases, and for obvious reasons. And that is, of course, mostly bacteria and viruses. So the whole point is that the concept, it's just done with different sequences. That's all.
So first of all, thank you for a great presentation. When listening to all the numbers, the number of species, etc., I couldn't help thinking how hopeless the problem you're working on is and trying to solve it. But the thing that struck my mind is, where do you reach the point of diminishing returns? Like in Costa Rica, your field workers, In that one picture, you said there are 1,000 species out there. So every plant, every butterfly, they're sending back, you're able to catalog because you've not cataloged it. But after a while, every sample that comes back might be the same sample that comes back. You already have the DNA. How do you get to the species that is very hard to find, that people don't see, that's in some remote corner of the earth?
There are a couple of points that are relevant, I think. First of all, I guess what we're most interested in developing is an identification system for organisms that you actually do encounter. So the ones that you don't encounter aren't such a problem. It would be nice to just be able to identify the things you do find. The second thing is the strategy for the first 500,000 species is to focus this effort on a rather small number of groups. So when it comes time to terrestrial life, we will try and be very comprehensive with butterflies and moths, with ants, with earthworms, but many other groups of life won't be primary targets in the first round. So we will build up species coverage, but there will be whole domains of life that are sitting there waiting for phase 2 and phase 3 of this project. And I think the other point is, as sequencing costs drop, as technology advances, it's going to be possible to scan immense numbers of organisms for the cost of the first phase of this project.
Let me add more to what Paul just said. As a person, I spend my life finding those last ones that were just referred to. And the fact of the matter is that when you get the sequence, you get more than the sequence. You get the natural history information that went with the thing that gave you the sequence. So you begin to build a set of information that's collateral to the sequence. So you build these natural histories. All of us who do field sampling quickly begin to see, all right, we've got the natural histories for the things that live here in streams, but we haven't got the ones that live in marshes. So now we stop sampling the streams and we go sample the marsh. Then we got the ones in the marsh, but we don't have the ones in tree holes in the tops of trees. So now we go up the trees and get them there. And you work your way through the world getting all the different pieces. Yes, there are a few species that may take like forever to find, but if the barcoder's in place, when you put one of those new ones in that nobody found, it's going to say to you, oops, this is not in the library. Would you please take a photograph of it? Will you please send us the collateral information that goes along with it? Would you collect it? All this sort of stuff. That the rare things that aren't found first, but eventually do get found, those will get in the sample as well. But when you're doing sampling to get the library, you break the world down into pieces, and you don't just wander around and get 10 million of these and one of those. And you do what we call stratified sampling across the system, and you gradually get more and more and more, a higher percentage of what's out there.
Do you guys have sort of a kind of a graded timeline of how you would build various kinds of barcoders with the technological milestones that you need to achieve in order to get the next level of miniaturization kind of worked out? And you know, what you have a barcoder in a building right now, and you want the barcoder in a pocket. How about a barcoder in a minivan? What's the gradient?
Yeah, what are the steps in moving from technology today where we need a room full of equipment that costs perhaps $2 million to Dan's $1 back pocket barcoder? And I'm sure you're correct if we look at most other technologies, and certainly cell phones would be a very good example, weighing a few kilograms, costing a few $1,000s in 1983. But performing basically the same service as a modern cell phone that weighs 76 grams and costs nothing, except a subscription. I think we will see the same transition. I am sure that we will see a desktop integrated device, and that will be phenomenally useful to people at border points, phenomenally useful to biodiversity scientists, phenomenally useful in educational settings where students move into the field to do field work courses. So yes, my guess is 5 years from now, sadly, we won't have the back pocket device, but maybe, Dan, do you want to be bolder and predict back pocket in 5 years?
I'll predict, the way I see things going, but I'm pretty naive, I guess, is I see the back pocket in 5 years, because I think what will happen is that when you get the one that'll fit in the backpack, it won't take somebody very long to say, you know, I could make my $1 billion for my life, me personally, my selfish life, by inventing the one that only costs $1, because then I'll sell 7 billion of them, and everybody will lose one every 3 weeks, and they'll want another one. It's like razor blades or like ballpoint pens, the sort of thing that, you know, there's just a dillion of them out there. And I see that's the direction it's finally going to go. And the world is going wireless, literally. I mean, the Costa Rica that I live and work in now is going wireless. The country is going wireless. So in a very short time, you won't have to carry all the damn library around in your comb. It'll just read off of a signal and be connected to you guys like that.
What kind of challenges do you see in regards to like open access to the information as opposed to having it be proprietary and sold?
Our concept is that the barcode is public domain. Like words, like English words and Spanish words and so on. And then what people want to do is take that and build it into various package formats for various people's specialty uses and all that. That's fine, that's proprietary, just like we do with words today. But for us, the barcode, the basic word, is public domain.
So in addition, from this little snip of mitochondrial DNA, can you get more information such as evolutionary relationships between organisms, or is it really just an ID tool?
Yeah, that's a very, very interesting and important point. And it is extremely nice to say that we gain a lot of additional information about the array of organisms that you're dealing with. So as an evolutionary biologist, I'm fascinated by the fact by carrying out these large horizontal surveys, we don't just get an identification, but we get a sense of the age of the species involved. We get a sense about the factors. This gene is a sentinel to everything that's going on in the mitochondrial genome and often in the nuclear genome. So you learn about rates of evolution. And of course, the other thing we get through the barcoding process at the end of the day is a total DNA extract, and we plan to conserve these with care. So when individuals decide to do tree of life studies that will undoubtedly require additional genes, that DNA extract is there. So we're working on very close and friendly terms with tree of life projects that want to burrow down and look at deep relationships. Barcode of Life is really decorating that tree of life with its leaves.
But let me add to that too, in that when you, as a user, okay, I send all these sequences up to Paul's processes, and they come back to me in these big trees. These big trees that show this is one thing, and this is another, and this is another, and this is another, when you get used to reading these tea leaves, what you discover very quickly is that groups that we classically call related end up grouping together. They have similar barcodes. And so that what happens is that there's a lot of what we call phylogenetic signal in them, but you can't rely on them 100%. But they tell you a lot in a signal sense. They say, oops, this thing is up here, and morphologically it ought to be down here. So it causes you to go back and look at this one that's up here, barcode-wise, and see that maybe morphologically we goofed in putting it down here. Sometimes that's the case, sometimes it's not the case. But there is signal, but you can't rely on it, which is one of the pushback we get all the time from the phylogenetic people who deal with genomes, saying, yeah, you can't use it. We showed 20 years ago that you can't use one mitochondrial gene to construct a phylogenetic tree. True, you can't. If you're a scientist, you want everyone to be in the right place, you can't. But it's probably 90% of that tree will in fact be where you get it if you use 15 genes. Next one.
One of the interesting things is you add more and more species to an analysis, single gene analysis like that. Character sets that were not phylogenetically informative become phylogenetically informative. So there's actually a great value in increasing species coverage.
Do you have a feel for the loss rate and the production rate of species as a function of time during, say, the last million years? Or
Yeah, that's a very interesting point. Well, the fossil record suggested that most species had a lifespan, marine species had a lifespan of about 4 million years. And those deep divergences that you could see in the trees, we can apply a molecular clock to those and estimate from the sequence divergences how old those species are. And the answer is most of the species that we live with have been fellow travelers with us for millions of years. So species are old. There are some settings where we can show that species have radiated explosively. The African rift lakes, the cichlid fishes, in a matter of 10,000 to 50,000 years, hundreds of species have been born. These are complex cases for barcoding, these very, very young species, but that's a rare situation. Most species are old and worth preserving, I would argue, because of that.
I know you said that you currently aren't working on prokaryotes and viruses, but is that something you'd be looking to do in the future, or do you feel like there are enough efforts going towards that with like the international nucleotide sequencing database collaboration, GenBank, and all those other groups?
There is a huge amount of money being directed towards prokaryote genomics, and we really feel it would be mission creep for us to try and move into that turf. We think the job before us is quite a substantial one, and mobilizing the workforce and the resources to do this job well will keep us occupied, and we'll look forward to colleagues pushing forward on the prokaryote front.
But you are correct to indicate that, because there needs to be a joining of these two highways somewhere not too far down the road. Because every time we put a leg into Guelph or into a tube in a freezer, whatever it is, of course, it's got viruses in it. It's got bacteria writing on it. And every bug has got a gut full of them. They're there. And at the time we go to the effort to get these things, to get their barcode, our barcode sequence, there's no reason at all why that same effort shouldn't then result in the material that the bacterial and the viral people get their raw material from. And these very same plants that we're barcoding right now in Costa Rica, we're doing, in fact, a plant virus barcode inventory of them.
Forgive me if I missed something, but every time you do a barcode reading, you have to pluck a leg or something and ingest it in order to get the reading on the species. I just have a question about scale on this. If you have 7 billion people on the planet who have one of these things and are plucking legs or wings from creatures, doesn't that begin to interfere kind of with the biodiversity on the planet over time? Or, I mean, no. There's just too many of these creatures to make it a problem.
That's right. There will be a higher incidence of 5-legged ants than there are now, but no.
Okay. I mean, I'm just asking the question, because you get 7 billion people doing something, that's going to have an effect, you know. If you're potentially putting something in the hands of 7 billion people, kind of what is the outcome of that? That's all.
Well, there's two kinds of people doing it. One kind is collecting the sequences for the library.
Right.
And that's, of course, a trivial amount of sampling.
Right.
And then there's all the people who actually then have a barcode in their back pocket. So what do they do today? All right, there's a beetle eating my bean. I'm in Nigeria, and there's a beetle eating my bean plants. Well, I kill it. I nuke it.
Right.
All right, you pull the barcoder out and you take a leg off one beetle and you stick it in there and it says, oh, that's such, and that's not going to affect your bean plant at all. Or this says, oh, wait a minute, yes, you shouldn't have this eating your beans and you better spray now. You know, yes, there's going to be a reaction, but we have that reaction right now. It's just that we don't have the accuracy that would go with this.
Well, I think what I'm saying is that at some level you're creating a new toy. And so people may be using it not to find out the beetle that's eating their beans, but just, you know, because you can.
Yes, that's true.
You know,
and we do that with reading too. So I mean,
but there's a down. I'm sorry, with reading.
Yes, exactly. You take second graders and you teach them to read. You don't ask, what are you going to do with the capacity of being able to read? And some of them will use it capriciously. Some of them will use it to damage the world. But most of them will, in fact, get
they'll be interacting with inanimate books, basically, and then maybe acting in the world. But here you're interacting with sort of, the animate world.
It's certainly an issue that has concerned us, and when you start talking about vertebrates, tearing bits off of a frog to identify it is something that would definitely be very unfriendly, and it's not necessary. You simply touch the organism, and for there's absolutely no question, if you simply stroke an ant or a caterpillar's skin, you will get more than enough DNA to identify it. And I mean, at the moment, the bug's on a pin, taking a leg off it.
Yeah.
It's very important we have these voucher specimens, but when people start to go out and do this in a naturalist context, I don't see them necessarily pulling organisms apart. That's the beauty of PCR. Okay. It needs almost no material.
All right, that feels better. Thank you very much.
Good. Yes?
Yeah, do you anticipate any legal issues as you go around sampling things such as Craig Venter ran into when he did his seawater sampling around the world and was arrested in numerous countries for taking samples until he signed away certain rights to what was discovered from those samples?
Do we expect any legal problems arising from the Convention on Biological Diversity and moving specimens out of countries? And the answer to that is we're building a deeply international coalition. This is a project that can't be done without international collaboration. And in some nations, the Costa Rica model of material moving to Out-of-country analytical facilities will not occur. In India and Brazil, analytical facilities within country will carry out the analysis, and the results will be deposited into a web-based system where the data is reposed, but the specimens stay within country. We will certainly operate within the boundaries of the Convention on Biological Diversity.
And since the Craig Venter sampling was brought up as an example in the Costa Rica that I work with, when he came to Costa Rica and took his marine samples, the Costa Rican government was exceedingly upset with that, exceedingly. This same government that has written into my collecting permit that you may collect samples for DNA barcoding anytime. Because everybody recognizes that this little snippet of mitochondrial DNA has basically no commercial value. And so the government has gotten to a point where it recognizes this is something that we don't care. It's a public domain. We're not worried about it. So I wouldn't care whether the sample sequence is taken in Guelph or whether we do it in Costa Rica or wherever we do it. It doesn't matter. That's the level of understanding of biodiversity genetic stuff, which we're beginning to see emerge in many, many tropical countries. It takes a while. It took us 20 years to get there in Costa Rica. But the Venter method was, in fact, not a happy circumstance.
Thank you very much.
Pleasure.